Thank you all for being here and thank you for the opportunity to speak today. I'm Sarah Shelton and I'm a postdoc at MIT and also at the Dana-Farber Cancer Institute under the supervision of Dr. Roger Kam and Dr. David Barbie. So today I'm going to tell you a bit about my project that really is involving vascularizing patient tumor microenvironment models in order to do immunotherapy studies. So the overview of our approach here is to develop patient-derived models by taking patient surgical tumor resections and then disassembling that into the different cell types that we need and then reassembling them in our model. So starting with taking a surgical resection from the OR, we mince it into small pieces and then sequentially filter it first through a 100 micron filter and then through a 40 micron filter. And so that gives us 3 different size populations of cells and spheroids coming from this single patient sample. So first, the intermediate size fraction from 40 to 100 microns, those are our PDOTs, or our patient-derived organotypic spheroids. They still retain tumor cells, stroma, extracellular matrix, and immune cells. So we call them organotypic spheroids instead of tumor spheroids. And so the Barbie lab at Dana-Farber in conjunction with Cam Lab has already done a lot of work characterizing the PDOTs and also comparing them to MDOTs, or murine-derived organotypic spheroids, and looking at response to immunotherapy, immune checkpoint blockade by doing live dead staining. We've looked at the populations of immune cells in the PDOTs relative to the primary tumor and found that these immune populations match. And so I really wanted to take these PDOTs and then put them in a vascularized model. So I think for doing immunotherapy studies, it's great if you can put your PBMCs or your T cells with your tumor spheroids, but I'm really interested in the extravasation and the role of the vascular biology in this process. So we need T cells, and I'm getting that from the surgical resection as well by expanding the T cells in that single cell small populations using IL-2 to expand the tumor infiltrating lymphocytes. And so then we also need the cells in order to create the microvascular networks. So the large size fraction that's not the immune cells or the PDOTs, I take that and further enzymatically digest that, and I'm getting out of that the cancer-associated fibroblasts. So that's the stromal cell population here. And then originally we were aiming to use the tumor endothelial cells, but we weren't able to isolate those in sufficient numbers. So we went ahead and started using the HUVEC in this model. And so I'll show you what happens when we start to build up this model from these individual components into the sort of full tumor microenvironment model. So if you're not familiar with our methods for generating microvasculature, we use the vasculogenesis model, and it works pretty simply by combining endothelial cells and stromal cells, typically fibroblasts, in a fibrin gel, and the fibroblasts support the connection, elongation, and luminization of the endothelial cells into perfusible microvasculature over the course of about 4 to 7 days. So the perfusible microvasculature ranges in diameter from about 2 on T to 200 microns. And we can co-embed tumor spheroids into this model at the time of seeding as the microvascular networks are forming, or you can preform microvascular networks and then add tumor spheroids to a port in the model. So the microfluidic devices for this model are very simple. So you need a single channel for your hydrogel containing the cells. That forms the microvascular networks. And then we have 2 flanking side channels for fluid flow in order to support the culture with tissue culture media. Or once the networks become perfusible, that's how we introduce T cells and have them perfused through the system. So since I am using cancer-associated fibroblasts in this model to generate the tumor microenvironment mimicking model from these patients, I think it's worth discussing a little bit what a cancer-associated fibroblast is and where they've come from. So the sort of most simplified version of what a cancer-associated fibroblast, or CAF, is that it's a normal fibroblast that's been activated. And so typically the most well-known mechanism of activation is TGF-beta secreted by the tumors, but there are also a number of other inflammatory and mechanical signals in the tumor environment that can activate fibroblasts and produce those CAF. So calf from different tissue and from different patients are heterogeneous. There's definitely a number of different calf subsets, and they are often categorized as myofibroblastic calves, inflammatory calves, even antigen presenting calves. So what this means is there's not really a single calf marker that you could sort of pick out and identify this is definitely a calf. But they should definitely lack epithelial, endothelial, and immune cell lineage markers. And they have elongated fibroblastic morphology, but you want to make sure they don't have mutations so they're not EMT cancer cells. So again, several of these markers exist. They're not exclusive, but they are useful to sort of confirm that you are looking at calf rather than normal fibroblast. So PCR on average on this tumor cell cancer-associated fibroblast population found that there's no big change in vimentin expression when I compare that to the normal fibroblasts. And vimentin is a sort of a pan-fibroblast mesenchymal marker. But there are upregulation in several calf markers, including fibroblast activation protein, caveolin-1, alpha smooth muscle actin, and fibroblast specific protein. So here it's worth mentioning that the normal fibroblasts that I'm comparing the calf to are not from the same patient. We didn't have enough tissue to sort of isolate normal versus calf from a single patient, but I'm using primary fibroblasts from healthy donors. And so most of my cancer specimens are from head and neck cancers. I have several oral cancers, or oral cavity cancers that will be discussing, and also thyroid and a subset of lung cancers. So in order to use matched primary fibroblasts, I also have lung, thyroid, and dermal fibroblasts to compare to. So starting off building this model, we were sort of building it up piece by piece and began with building microvasculature, combining the HUVEC endothelial cells with the cancer-associated fibroblasts. So now I'm going to be showing you maximum intensity projections of the microvasculature in these models. And in this case, these images are perfused with green fluorescent dextran, so you can see which segments of the microvasculature are perfusable. And so right away, it was really obvious that there was a lot of variability from patient to patient. So everything was seeded in identical conditions and cell numbers, and we see really broad differences between patient to patient calf and how they support the morphology and the generation of microvascular networks. So you can see from one extreme in the top left, where we have sort of minimally perfusible networks, to the other extreme in the bottom right, where we have really wide flattened networks that have sort of lost their cylindrical cross sections. But the majority of the samples lie somewhere in the middle. So I took a subset of all my patient-derived fibroblasts, and I compared that to the normals, and I started looking at different aspects of the vasculature, including the morphology and the permeability. So to look at the geometry, I used a program in MATLAB called AutoTube in order to mask and skeletonize the vessel projections in 2D in order to quantify the mean diameter, the number of branches, and the branch length. And so there was a significant increase in the mean diameter in the calf networks compared to the normal. It was a small increase of about 5 microns. And additionally, a significant decrease in the number of branches, while an increase in branch length. And so from the morphology parameters, we can also get functional parameters such as permeability by, again, perfusing with the fluorescent tracer like dextran, and then measuring the change over time of the intravascular to vascular to extravascular signal intensity of this dextran. So here I'm showing you 2 maximum intensity projections from the normal fibroblasts, since I haven't shown those yet. So here's a typical image of a lung fibroblast network and a thyroid fibroblast network from healthy donors. I haven't shown you the dermal fibroblast networks because in the identical conditions to form all these other networks, those networks were not perfusible. So in the normal lung and normal thyroid fibroblast networks, permeability is fairly low. and pretty consistent. However, when I compared across fibroblast types, there's a lot more heterogeneity, a lot more variability, and also higher permeability. And so this is in line and consistent with the literature about the permeability in the tumor microenvironment and enhanced permeability and retention effect in tumors. And this is due to the fibroblasts alone, rather than the typical thought that the tumors are secreting VEGF and contributing to permeability. So yes, as you might imagine, we're wondering why these fibroblasts are promoting differences in the vascular morphology and function. And so our first thought was VEGF, vascular endothelial growth factor. It's a pro-angiogenic factor, and we wanted to look for that in the system. But we were also curious of what else these fibroblasts might be secreting that would be impacting morphology and permeability in these vessels. So I did a Luminex assay and cytokine assay for looking for angiogenesis markers. And I think we're frozen. There we go. I also simplified the model in order to look at sprouting angiogenesis. So a lot of these features had already pointed to this pro-angiogenic phenotype as we expect in the tumor microenvironment. So I simplified the model down to the hydrogel with the embedded fibroblasts and now localizing the endothelial cells as a monolayer on one side. So as we apply the monolayer, the endothelial cells will then sprout into the fibroblast-laden gel, and then I can image the fluorescent fibroblasts and do image processing to crop and segment and quantify the degree of sprouting in each of the different conditions. So it's really apparent right off the bat visually that the CAF are promoting much more sprouting than the normal fibroblasts. And so we see longer, more developed, more abundant sprouts in these CAF models than the normal fibroblasts. And when we quantify this, so I quantified sprout area, density, and length, and I found significant increases in all three, ranging from about 1.5 to 2.5 fold increases in sprout length, sprout area, and sprout density. And so again, we think that this is probably related to VEGF, and the cytokine array reiterated that fact that VEGF, the vascular endothelial growth factor, is about 5-fold increased on average in the cancer-associated fibroblasts over the normal fibroblasts, again, with some heterogeneity between the patients. And so again, we would expect VEGF to show up in this screen. It's expressed by cancer cells, fibroblasts, macrophages, a lot of the stroma. It's really well-known factor in the tumor microenvironment to play a role in angiogenesis, typically in response to the hypoxia that occurs in the tumor microenvironment and the HIF-1 alpha signaling. And of course, there are several FDA-approved drugs for VEGF and the VEGF receptor approved for combination therapy in oncology. So as the title of this slide hints at, the other factor that we found was CCL5, and this was a little bit less expected. CCL5 or RANTES was increased just over 2 folds change from the normal fibroblasts, and it's typically a T cell chemokine. It's expressed by activated T cells, although in the literature it's also known to be involved with angiogenesis, typically through VEGF stimulated angiogenesis, but also related to endothelial cell proliferation and migration. And then there's also a great deal of literature about its effects directly on tumor cells and in response and making them more responsive and to tumor progression and metastasis. As we saw, VEGF and CCL5 were both implicated in the system and highly expressed by cancer-associated fibroblasts. I started looking at whether or not we could tease out their different effects in the sprouting population in fibroblasts. So now I'm using cancer-associated fibroblasts and reminding you what those control sprouts look like here. And then if we block CCL5, we see a really noticeable reduction in the length, the number, the density of the sprouts. And then furthermore, if we block VEGF, it becomes an even greater reduction in the sprouting. And it looks pretty similar when we block CCL5 and VEGF simultaneously. And so this is all using neutralizing antibodies and a research grade bevacizumab VEGF. So quantifying this data, looking at 4 different cancer-associated fibroblasts and looking at the sprout area as our indicator of sprouting. And we see reductions from the control conditions to all of the conditions, anti-CCL5, anti-VEGF, and the combination of both. And interestingly, if you look at the blue and the orange bars, those are CAF1 and 3, and those had the highest baseline production of CCL5 in my cohort. And that's where we see the greatest reductions in sprouting when we apply the anti-CCL5 treatment alone. So as expected, when you block CCL5, it's most effective if the cells are already secreting quite a bit of it. The 2 groups where there was no significant difference was anti-VEGF treatment versus anti-CCL5 plus VEGF. So this sort of reiterates that VEGF is the dominant effect here, but that CCL5 is probably having a synergistic effect in the tumors that's producing this great amount of sprouting compared to the controls. So we started thinking more and more about, like, why are we seeing CCL5? And then we looked through the other factors that were increased in the cancer-associated fibroblasts. And we're thinking about, like, a lot of these factors are overlapping in between promotion of angiogenesis and inflammation. So CCL5, I've already discussed, is a T-cell chemokine. However, it also is known to recruit monocytes. And then CCL2, or monocyte chemotactant protein, was also high on our list of hits. And as it sounds, it's a monocyte recruiter, but it also can be used to recruit T cells. And as we look through the other markers in this list, and things like Ang2, CXCL4, and placental growth factor, and started looking into the literature, what really sort of popped out is that we were sort of looking at a dichotomy between T cell and monocyte recruitment. And so since we have perfusible microvascular networks, that's something that we can test directly. So we started looking at how angiogenesis and inflammation are connected in this tumor microenvironment model that's created using the cancer-associated fibroblasts. So in order to quantify immune cell recruitment in this model, we label primary monocytes and T cells with different color fluorescent dyes. In this case, this is from blood from healthy donors. And we perfuse equal numbers of both cell types, perfuse them for an hour under gravity-fed flow, and then wash out the unbound cells before quantifying the retention and relative abundance of each cell type in these models. So here are the images, maximum intensity projections of typical microvascular networks with normal fibroblasts, or microvascular networks generated with CAF, with the same endothelial cells here from the HUVEX. And the monocytes are labeled in magenta and T cells are in green. And so you can see in the normal fibroblast networks that the number of immune cells is relatively low. We haven't treated this with any inflammatory cytokines in the media. It's just the contributions of the 2 cells in this fibrin gel. And on the other hand, when the CAF are in this microenvironment model, we see a lot greater infiltration of the monocytes in pink and the T cells in green. And when we quantify the numbers, both monocytes and the number of T cells were significantly higher in the CAF models than the normal fibroblasts. And interestingly, in both normal and CAF, we see higher numbers of monocytes compared to T cells in both model types. So getting back to the holistic model that we're trying to develop to have patient-derived models of the tumor microenvironment, we really want to be able to do immunotherapy studies on personalized patient models. So we have the CAF, we have the tumor spheroids, and we have the T cells. But again, if we want to use the microvascular networks, we really need an endothelial cell, and we need an endothelial cell model that's not going to conflict with our T cells and produce a graft versus host response. So we could think about doing HLA matched endothelial cells and have a library of endothelial cells to match for each patient. But we took a different approach, and we were looking at trying to make the endothelial cells invisible to the T cells. So T cell recognition is the T cell receptor binds to the MHC peptide complex on the target cell, and the MHC class 1 has one fundamental building block, which is the beta-2 microglobulin, or the B2M protein. And so we knocked that out in our HUVEC cells in order to make HUVEC that would be invisible to these CD8 T cells. So we knocked out B2M from the HUVEC and from healthy fibroblasts in order to make immune invisible microvascular networks. And so we knock out B2M, and then they don't express MHC class 1. So we can form really beautiful microvascular networks with these HUVEC and fibroblasts that don't express MHC class 1 via the knockout of B2M with CRISPR. And so that is going to allow us to combine these fibroblasts, healthy fibroblasts in these HUVEC with any of our patient models. So we have sort of a universal HUVEC now to go with all of our models. And so the full model picture is to combine these B2M knockout HUVEC either with our cancer-associated fibroblasts or with our B2M knockout fibroblasts. And we could also combine those two in different ratios to look at the impact of the CAF on the microenvironment. And when we combine those endothelial cells and stromal cells, that's what generates our microvascular networks. And within those microvascular networks, we're embedding the PDOTs, those patient-derived spheroids. And then after about a week of culture, we have perfusible networks, and then we can flow the T cells through the system in the presence or the absence of a treatment such as the immune checkpoint blockade. So the overall goals of this are to do long-term recirculation of these lymphocytes in order to image their extravasation and migration through the tissue to the tumor spheroids. And then downstream of that, with treatment and with and without treatment, we want to do cytotoxicity monitoring of live dead. And we can also sample media from the system to do cytokine and look at increases in things like granzyme B, TNF alpha, interferon gamma. So the model is still in development, and we're building it in the piecewise fashion. So on the left side of the screen, you're going to see melanoma, patient-derived melanoma spheroids labeled in green. These are in a microvascular network generated with those B2M knockout HUVEC and B2M knockout fibroblasts. And so you can see the microvasculature forms nicely around the small spheroids. And then panels B and C on the right, we have microvascular networks formed with the B2M knockout HUVEC and in combination with cancer-associated fibroblasts from one of the patients. And then I've perfused T cells from the matched patient sample. And so T cells in this model flowed through the model. They adhered to the endothelium, they were trapped, and they extravasated. So in the panel on the far right, you can see the T cell in red extravasating from the microvasculature, and then one farther down in the image that has already migrated into the interstitial space. So in conclusion, I want you all to see that the cancer-associated fibroblasts support vessel formation in this vasculogenesis model. And this resulted in slightly larger diameter vessels, changes in the number of branches and branch length, as well as increased vascular permeability to 70 kilodalton dextran. And so while we saw a range of geometry and phenotypes in these cancer-associated fibroblast networks, most of them live somewhere in the middle here with nicely perfusible vessels that look quite similar to our normal vessels. So these differences are probably due to the secreted factors expressed by the cancer-associated fibroblasts that induce both angiogenesis and inflammation. There seems to be a lot of crosstalk between those two. And the CAF resulted in much greater sprouting than the normal fibroblasts, which was able to be inhibited by blocking CCL5 marginally, and then more completely by blocking with VEGF or VEGF and CCL5 in combination. Finally, the CAF networks retain more of these immune cells, as we saw in the images and quantifications there. And so as we put all of these model pieces together, we're really trying to build these patient-specific models of the tumor microenvironment, where the microvasculature is a fundamental component in order to model T cell extravasation, migration, and homing to these tumors, in order to model more of the components and the steps that are involved in response to immunotherapy, and then hopefully to be able to look at patient-specific responses to immunotherapy in these models. I'd like to thank the CAM Lab and the Barbie Lab at Dana-Farber and all of my collaborators.